In today's video, we are going to answer the question, does soil microbiology actually work? This is something that I talk a lot about in other videos. I always kind of go back to this idea that if we want to grow the healthiest, tastiest food possible, we need to maximize the life in our soil. So in this video, we're actually going to focus on that question and we're going to test it. And I'm really excited to show you this. It's taken a few weeks to get this far. It's not a spectacularly big trial that we've done, but it's taken a little bit of time to allow things to grow. So for all you doubters out there, or for anyone kind of sat on the edge, does soil microbiology work? Stay tuned because the results are pretty amazing, I think. Now, as I've said, this has taken a little bit of time to not only set up, but actually to get the results for. And in fact, as I'm recording this at the moment, there's still a couple of results that I'm waiting for. So what I've done is broken this trial and this video up into a small series. There'll probably be about 2, maybe 3 videos in total. And I'll go into the process, exactly what I've done, how I've done it, in as much detail as I think you guys can handle. And hopefully at the end of this, you'll have a simple kind of trial that you can carry out yourself if you want to test your own soil and get an understanding of the effects on what food you might be growing in it. So if you like videos that give you tips, that talk about soil health and show you ways and principles that you can apply yourself to grow the healthiest, tastiest food, do give this video a like and consider subscribing to the channel. And without further ado, let's get stuck straight into it and look at exactly what I did. So for this, I was working with a bag of soil that we'd already tested. So I had some baseline measurements for where the microbiology was. You can tell it's an old sample because I split the bag multiple times, making a mess everywhere. But this is just a normal soil, untreated. I'm kind of mixing it up here and splitting it between 2 bags. You can see I'm actually having to change the bag that it's in here because I made such a mess of the first one. And the idea is to kind of mix it evenly throughout so that I've got 2 portions of soil here that you can see that are as close to the same as possible. And what I'm just doing now is checking that I've got approximately the same amount of soil in each bag, because ultimately what I'm going to do is pop this up. So just done the first bag, doing the second one here. One of these is going to be split down and kind of treated so that we can minimize the soil life that's in it. So always talk about labeling your sample bags. So I've put one lot of the soil into one of the bags, which I've called Alive, and then the second bag I'm going to put into a beaker, which I'm doing here. And the idea of this is that it means that we can treat it, and there's quite a lot of soil in this. I think that's approximately a litre from memory. So it's completely filled, and actually you can see in the end I had to put it in 2 smaller glass beakers in order to fit them in the autoclave, which is over there. So here we go. I'm going to pop them in the autoclave, which is in essence like a pressure cooker. The idea of this is to kill off as much of the soil life as possible, so that I have 2 batches of soil, one which you saw labelled Alive, and this one which I'll probably refer to as sterile, but actually it isn't sterile, it just has significantly reduced microbiology within it. So having got my soil ready, I then went on to pop things up ready for the trial, which I'm going to do now, and you'll see in super duper lo-fi because I recorded it all on my phone. So these are the materials that I used. I had some pot trays here. I then got some 10.5 centimetre pots, which I had cleaned in the sink with some antibacterial dish soap and then rinsed them thoroughly. I got 6 packs of seeds. These are cress. I'm using these because they'll grow very quickly, and then we can get this video out to you guys and see the results. This is my bag of natural soil, so untreated. It's hopefully kind of as alive can be in terms of microbiology. This is one of the beakers, here's two of the beakers of my autoclaved soil, which is less active. I'd like to call it sterile, but in reality it's not. And then most important piece of kit when you're doing tests and sampling is a Sharpie pen. So what I'm going to do now is label up the pots. With the Sharpie, we're calling the alive soil kind of natural, so we're giving these an N to show which pots are which. I'm doing 3 replicates of each, so 3 alive and 3 less active or sterile, which we're labeling with an S. It's not the most scientific process, but I think the 3 pots should hopefully prove without any doubt that even though we're not using scientific kind of rigor levels here, that there are noticeable differences. So quick time lapse showing me filling the pots up. This was indeed an untreated kind of almost natural soil. You can see I'm having to pick lots of bits of old organic matter detritus out of the pots to kind of get them kind of as level as I could. And so I'll pop the first lot of natural samples onto their pot trays, and then using the beakers here, which I kind of kept the lids on, some waxed kind of film lids to keep them as free from microbiological contaminants as possible. So there's my pots ready. Here they all are. I have kind of lined them all up. You can kind of see that the soil levels are roughly the same. I try to get them as even as I could. You see where I'm kind of pointing there are pretty similar in terms of general appearance here, which they're all sort of lined up now ready for adding the seeds, which I will do now. So another quick time lapse. In order to kind of make this as consistent and as fair as possible, considering this isn't a scientific trial, I basically am using 1 packet of seeds per pot, and I'm distributing these as evenly as I can just across the surface. We're not going to cover them over, we're just going for as good a coverage as we can possibly get over the tops of the soil. And ultimately I don't think it's going to matter too much in the end, because the measurements that we're looking to make at the end of this will be how much they have grown. So we're going to do that with a test of organic matter. Maybe we will look at nutrient density in a very simple way, a system called refractometer, which looks at the bricks, and we'll do some soil assessments as well. So now that they've all been done, here we go. Quick look at the pots, the natural ones on the bottom, the less active across the top, and you can see that they are pretty evenly distributed. That's a little bit perhaps lopsided, but I've done the best I can to keep this as even as possible. This is the exact type that I use, just simple cress, curly cress that I bought off of Amazon. I followed the instructions here, which is just to scatter directly onto soil, which it kind of says to do if you're doing this outside. If you're growing inside, it tends to be on tissue or cotton wool. And what we're going to do is put them to one side now and just monitor them as they hopefully grow over the next few days. So last time lapse before we get on to the growing, quick time lapse of watering these things up, which I'm doing initially with kind of a pump action spray, and it is literally just tap water. I haven't taken any particular care or treatment over the water types because I'm going to need to repeat water them throughout the week or so. So quick water over the top with a spray to not disturb the seed positions too much. I'll probably spray them again over the first couple of days, but one of the reasons I'm using the trays on the bottom is so that I can water from the bottom. Hopefully the soil texture will lend itself to good capillary action, so they'll be able to get the majority of the water that they want from the bottom as well, once kind of water levels increase sufficiently. So we'll pop them to one side now. This is where they're going to grow. They're getting some reasonable light actually under a skylight in my extension. So it's pretty even. There's no particular drafts or heat sources to one side. So we're just going to keep an eye on them now over the next few days and see if we can notice anything. So now that you've seen them all potted up where I've got them ready for growing, let's have a look over the next few days at what I see when I go and check them out. So this is after a day and a half. See, they are starting to germinate. Additionally, and this is of interest, I did notice that these three, 1, 2, 3, are the natural soil, so they're active, whereas that and those two are the kind of sterile, but not quite as sterile. Must say, I do see quite a difference, actually. I'm surprised at how much of a difference there is. We'll see how they develop and grow there. Okay guys, so this is really interesting. We're now about 2 days on. Have a quick look. These are the sterilised soils here. But interesting, you notice the more natural active soils in the middle. If I kind of pull back, you can hopefully see that actually the germination across the three of them, you can see green now, the more sterile soil, much lower germination rate. Yeah, I'll pull back, hopefully you can see. Pretty amazed. I wasn't actually expecting it to be quite as pronounced so early on, but yeah, look at that compared to, say, that. So just thought I'd do another quick update because there have been some interesting developments throughout the day. So this is kind of a nighttime update of, oh goodness me, I think it's day 2 or 3. They are my kind of sterile. That is my natural. That's a natural. That's a natural, and that's another sterile. I'll just kind of draw back. I think it's fairly clear to see that that, and that, which are the natural, are significantly greener and a lot more advanced in terms of their growth. You can kind of see them here compared to these ones. And I did mix up the positions, so in terms of daylight, it should be fairly evenly distributed, but perhaps tomorrow I might move them around just to make sure. So this is kind of the beginning of day 5. Things have kind of evened up a little bit, although I would say, and maybe the camera doesn't show it, that these 3 do appear a little bit darker. Maybe we can get a little bit of a close-up. No, it doesn't really do it justice, but we can see it kind of evened up a little bit, but looking pretty good. So also on day 5 I have now topped up the water in all of them with the exact same amount. There seems to be quite a good capillary action with this particular soil, and so I've kind of just watered them in the trays beneath. But one thing I did notice is that the natural soil, so that's kind of the biologically active, seem to have drunk significantly more water than the kind of slightly more sterile soils as well. Didn't drink all of it, but almost all of it, tiny little puddle left, whereas the kind of more sterile soils, there was quite a high covering of water across the bottom, certainly out of 2 of the 3. So interesting to see the water consumption is a little bit different between the samples as well. So this is day 6, and I think tomorrow I will be harvesting the cress. Ultimately I didn't change the positions. I thought I didn't want to disturb the conditions of the pots. So this is one of the natural, this is one of the not quite sterile, as that is as well. Natural, kind of sterile, then there's another natural at the back. You can see the difference here. Interesting to note is if I look at the bottom of the pots, so that's the kind of sterile, no roots. If I look at the soil biology, you can see much more vigorous roots. We'll look at this, which is a sterile, no real roots. Let's look at this one, which is a natural one at the back. And again, you can see lots of roots. In fact, I might as well do them all now. Here we go. This one of the kind of sterile, no roots. And the one with soil biology, big fat roots. So really quite a marked difference. So that wraps up part 1 of this video. In the next video, we're going to have a look at the results in a bit more detail. So I'm going to carry out a bricks test, which is a way to look at the nutrient density of the cress, and also I'm going to weigh the organic matter to have a look at the impact of soil life versus no soil life on the plants themselves. So stay tuned for that. If you want to be alerted when that video is released, do subscribe to the channel and click the notification button. So without further ado, stay tuned and I look forward to seeing you in part 2.